Greetings everyone, and welcome to another Genetic Engineering and Biotechnology News webinar. Our presentation today is entitled High-Throughput Glycan Analysis of Monoclonal Antibodies. I'm Jeff Bogaliskas, Technical Editor for GEN, and I'll be the moderator for today's webinar presentation. Protein glycosylation represents one of the most commonly observed post-translational modifications. The presence or absence of these complex moieties have a significant impact on the efficiency, pharmacokinetics, immunogenicity, and stability of protein therapeutics such as recombinant monoclonal antibodies. Historically, glycan analysis has presented unique challenges for researchers. Yet in recent years, technological advances have allowed investigators to develop new methodologies that maximize the speed and efficiency of glycan detection while increasing the overall yield of the desired glycosylated biomolecule. Let's hear from our speaker, who will inform us about a new method for the purification of monoclonal antibodies and how he incorporates high-throughput approaches in his research. Our presenter today is Dr. Ronnie Dosanjh, a senior scientist at B&D Life Sciences. Dr. Dosanjh will tell us about his novel glycan analysis techniques and how he has incorporated the Assay Map Bravo system to automate and improve his biotherapeutic manufacturing workflows. Before we begin, I want to encourage the audience to submit questions for our Q&A session at the end of the presentation. We will try to answer as many questions as we can, so simply type your question into the Q&A box on the left-hand side of your screen and hit submit. So, without any further ado, let's begin our webinar. Ronnie, the floor is now yours.
Good morning, everyone. It's nice to have this platform to discuss some of the things we are doing here at BD with regards to monoclonal antibody characterization. I would like to thank the organizers, Jen, for this opportunity to speak with you today. So today we'll be discussing a semi-automated high-throughput methodology for the characterization of monoclonal antibody N-glycosylation. I will start with a brief overview of BD Biosciences, and in particular, the Advanced Bioprocessing Division. We have a long track record of industry support in cell culture supplementation through our DIFCO and BBL brands. We continue to provide quality, media, and supplementation products for the biopharmaceutical market. This takes the form of commercial catalog media and supplement products, and also customized media solutions. To date, BDAB have been used in the manufacturing of more than 110 biopharmaceutical drugs in the human and animal health markets. I would like to review the aims of the work that will be presented today. So primarily, we sought to assess the Agilent Assay Map Bravo technology in an attempt to streamline the process of analyzing monoclonal antibody N-glycosylation. Being a liquid handling unit, the Bravo would allow for automation of certain processes, for example, protein purification. So the wider aims of the work presented here involve targeting an improved efficiency and increased throughput of analyzing multiple media and supplementation conditions in CHO cell culture. So the primary aim of this talk will be CHO cell culture and will be the focus of the work presented here. Before I delve into the main body of the presentation, I just wanted to review protein quality and what we mean by this term. Maintaining protein quality is of paramount importance to biotherapeutic manufacturers and ultimately the patients who receive such treatment. It can be impacted at several stages of process development, including clonal selection, cell culture, protein purification, and final formulation. Shown here is the typical structure of a monoclonal antibody, and on it are shown the typical covalent modifications that can occur. These modifications include pyroglutamic acid formation, deamidation events, where glutamine and asparagine residues form glutamic acid and aspartic acid residues respectively, methionine oxidation, and the C-terminal lysine variants on the heavy chain. The focus of today's talks are N-linked glycosylations. These are the black branch structures that can be seen towards the bottom of the heavy chain and often occur on conserved asparagine residues. So really, with this figure, I really wanted to emphasize here that there are a vast array of potential modifications that can occur on a monoclonal antibody, and what a complex task it is to control them. Those modifications that directly impact a biological therapeutics function are often termed critical quality attributes, and this is the term that I may come back to throughout the presentation. Within advanced bioprocessing, we seek to better understand the impact of our cell culture media and supplements on these monoclonal antibody modifications. As a general overview to the presentation, I'll be covering the following topics. Firstly, a summary of the previous methodology employed in the purification of monoclonal antibodies and for N-glycan characterization. This method had some bottlenecks, particularly with the protein purification, as I will describe later. The N-glycosylation portion of this protocol is based on the Waters GlycoWorks methodology that uses a fluorescent label called 2-aminobenzoic acid, or 2AB labeling. I will then move on to discuss our evaluation of the Bravo. We assessed protein purification yields in different sample matrices. These are the typical sample matrices that we encounter with our CHO cell culture media. Also, the protein binding capacity of the Bravo protein A cartridges were assessed. In terms of N-glycan assessment, we assessed the Prozyme GlycoPrep Plus Rapid N-glycan sample preparation kit. This kit is designed to be automated on the Bravo platform, and indeed the assay map has built-in software to utilize this kit. We made a direct comparison of the automated Bravo approach to the manual or benchtop 2AB labeling methodology. I will begin with the protein purification methodology that was previously adopted. This involved the use of GE high-trap columns on a GE Actor Explorer system. These are standard pre-packed protein A columns that are commonly used with these systems. The advantages of this approach include the purification of large amounts of protein, up to 20 milligrams binding capacity with a 1 ml column. And then there's the ability to view real-time UV elution profiles. There are several disadvantages to this method, however, particularly in the context of purifying multiple samples, as would be expected in a media supplementation screen. Firstly, it is time-consuming, with each sample taking approximately 1 to 2 hours to purify. This includes column equilibration, sample loading and binding, and the wash and elution steps. It's further necessary to strip and regenerate the column between samples to avoid potential cross-contamination. A further disadvantage is low throughput, since only single purifications can be performed. It's not possible to align several columns on the same system and purify them all at the same time. The typical elution volume obtained with a 1 ml column is up to 5 mL. Depending on the concentration and volume of the sample load, culture supernatant, the final purified protein may require concentration for downstream applications. In our experience, the purification of 5 protein samples with N-glycan preparation and analysis can take up to 5 days. So as you can see, there are several limitations to this approach with regards to sample throughput and general efficiency. Moving on to the N-glycan preparation methodology employed. The Waters GlycoWorks with 2AB labeling kit works through the following steps to achieve final purified N-glycans for analysis. Step 1 covers the denaturation and deglycosylation of the purified IgG. The IgG is initially denatured. The subsequent addition of DTT and iodoacetamide acts to first reduce the disulfides and secondly to prevent the reformation of disulfide bonds. Deglycosylation is performed with the enzyme PNGase F in an overnight reaction. Step 2 involves the purification of released glycans using a Hillick microelution plate used in conjunction with a vacuum manifold. The Hillick microelution plate can process up to 96 samples. The purified glycans are then fluorescently labeled with 2AB. This labeling reaction takes approximately 2 to 3 hours. Excess label is removed with a second round of Hillick microelution-based purification, shown in step 4, to yield the final purified and labeled N-glycan. The labeled N-glycan is then analyzed on a UPLC with a fluorescence detector with a Hillick-based separation column. It is also possible to perform LC-MS analysis with the labeled glycans or MOLDI-MS with the glycans extracted from step 2. I will now cover the evaluation of the assay map Bravo. A primary aim of the evaluation was to assess the protein A purification yields from cell culture supernatants. In particular, we were interested in any potential effects of the IgG sample matrix on purification yields. A CHO monoclonal antibody producing cell line was cultured in a batch mode with base medium, which is sample 1, as shown on the table, base medium supplemented with peptone as sample 2, and base medium supplemented with chemically defined or CD supplement as sample 3. Additionally, a purified human IgG sample in PBS was included as a control. Cells were harvested and supernatant collected for the subsequent IgG purification experiments using the Bravo. The Bravo head contains 96 barrels that can mount 96 individual protein A cartridges. Each cartridge is packed with 5 microliters of protein A resin. So the purification efficiency was assessed by the triplicate loadings of 25, 50, 75, and 100 micrograms of IgG containing cell culture supernatants alongside the human IgG control protein. The Bravo software called VWorks contains a simple and easy to use protein purification module. The sample load volumes and load flow rates can be adjusted as required, as can be the volumes of the wash steps and elution. For this experiment, the sample load volume used was 1 ml, which is the maximum possible load volume. The composition of the equilibration wash and elution buffers used were as originally suggested by Agilent. So at the end of this process, purified IgG was eluted directly into a 96-well half-area UV plate. This allowed for the direct measurement of protein concentration by UV visible spectroscopy. The protein concentration of the eluate was determined directly by UV visible spectroscopy. Quantitation was achieved by referencing an independent linear standard curve, shown here, prepared for the CHO monoclonal antibody and the human IgG control protein. The figure shown here depicts the calibration curve generated for the CHO protein of interest. So we have quite simply the monoclonal antibody concentration on the x-axis with the absorbance shown on the y-axis. So moving on to the results of the protein A purification of IgG in different sample matrices, which is shown here in the bar chart. The x-axis details the different sample matrices. So we have, moving from left to right, base media culture, base media and peptone culture, base media with CD supplement, and then the human IgG control. Each of the colored bars represents a different IgG load amount in micrograms, from 25 micrograms up to 100 micrograms. The purification recovery as a percentage is shown in the y-axis. The assay map Bravo purification recoveries obtained across the 4 samples ranged from 66% to 91%. The percent CV across all replicate samples ranged from 0.5% to 15.8%, with an average of 4.5%. For some of the samples, there is a slight trend towards increasing purification yields with decreased sample loads. So just to summarize, the Bravo was able to purify the monoclonal antibody of interest in a range of different matrices. We wanted to also assess the maximum binding capacity for the protein A cartridges. A quantitative capture or breakthrough experiment was designed where triplicate loadings of 125 micrograms, 150, 200, and 300 micrograms of IgG were performed. Protein quantitation, once again, was determined by referencing a linear standard curve prepared for the target IgG. So here we have the results of the quantitative capture experiment. The x-axis shows the microgram load of IgG, with the y-axis showing the microgram recovery of IgG. Up to 300 micrograms of IgG was loaded, and the maximum recovery of IgG approaches approximately 160 micrograms. It's clear that when we approach the maximum binding capacity, the efficiency of protein recovery is reduced. This is very useful information moving forward, as it allows us to saturate the binding of each protein A cartridge, particularly if we are not concerned with quantitative recoveries of protein. Next, we assessed the Bravo's preparation of N-glycans from purified monoclonal antibody. The Bravo VWorks software contains an easy-to-use N-glycan preparation module that was designed in conjunction with Prozyme for use with their GlycoPrep Plus Rapid N-glycan with 2AB labeling kit. Using the Bravo purified IgG from the 3 different sample matrices, N-glycans were digested and labeled with 2AB using the GlycoPrep Plus system on the Bravo platform. In parallel, the same purified IgG samples were prepared for N-glycan analysis using the same manual GlycoWorks protocol described earlier in this presentation. That's shown to the left. From this point on, I will refer to the Bravo prepared samples as using an automated approach and the GlycoWorks preparation as a manual approach. All samples, whether prepared manually or automated, were analyzed by a UPLC fluorescence methodology. So this slide outlines the main differences between the 2 N-glycan preparation methodologies. I've already covered the manual approach, which is shown on the left side of the slide. The automated approach has a few key differences which I will outline now. The denaturation, reduction, and deglycosylation can all be completed in just a few hours. This is achieved by combining the on-cartridge immobilization of the glycoprotein together with the deglycosylation step. The fluorescence labeling reaction has been optimized to last 1 hour. The Bravo elutes the labeled N-glycan samples in a small volume of water. Therefore, there is no need for an additional drying step prior to sample analysis. So just to summarize, the same glycoprotein samples were prepared using the 2 methodologies described and the purified N-glycans analyzed by fluorescence UPLC. So this slide shows a comparison of the fluorescence signal output obtained for the same sample, which is the base media culture IgG. The elution profiles were found to be very similar, and the identities of the eluted N-glycan species are shown. Peak assignments were made using either the Walters GPTS standard or with Prozyme's Glycor 2AB biantinary and high mannose partitioned glycan library. Over the next few slides, I will detail the results of the comparison assessment. I will do this by showing the results of each sample type. Just as a reminder, the samples are 1. Base media, 2. Base media with peptone, and 3. Base media with CD supplement. This particular slide shows the data from the base media sample. The x-axis shows the name of the glycan species, and the y-axis details the percent N-glycan distribution. This is calculated based on the area of each peak found in the UPLC fluorescence profile. The data for the manual preparation is shown with the orange bars alongside that for the automated approach, which is shown in the blue bars. The results show a strong correlation with the identified N-glycans between the 2 preparation methodologies. This slide shows the results of the base media and peptone sample. Again, the data show a strong correlation between the 2 preparation methodologies used. And here on this slide, we have the results of the base media and CD supplement sample. Again, the data show a strong correlation between the two preparation methods. Taken together, the data shows that we were able to achieve very similar results using the automated approach compared to our manual methodology. For both sample preparation methodologies, the percent RSD values obtained across all of the major glycan species was less than 5%. The next slide will show all of the data plotted together so that we can make some inferences about the effect of media supplementation on N-glycan distribution. As I mentioned in the previous slide, here we have all of the data plotted on a single chart. The blue bars represent the base media data. The red bars represent the base media and peptone data. And finally, the green bars show the base media and CD supplement data. The lighter shade for each color represents the manual methodology, and the darker shade represents the automated methodology. So when we look at the data, we can see that the N-glycans isolated from base media samples supplemented with either peptone or CD supplement have a similar profile. This is apparent when comparing the red and green bars, as they tend to be very similar. The unsupplemented base media has a distinct profile, as you can see when you compare the blue bars. And it's characterized by comparatively lower G0F levels. G0F is the highest peak or bar on this chart, and it's also characterized by higher G1F and G2F levels. This suggests an increased flux through the galactosyl-transferase pathway, which promotes the addition of galactose moieties. The unsupplemented medium is also characterized by greater levels of mannose-containing glycans. So just to summarize this particular evaluation, purification of the different IgG matrices was found to be robust with reproducible yields obtained for each condition. The quantitative binding capacity for the protein A cartridges was found to be in the region of 160 micrograms. The automated N-glycan purification approach showed similar results to that of the manual approach. And some further summary and conclusions. The ability to purify up to 96 different samples in a single day with the Assay Map Bravo has improved protein purification throughput significantly when compared to our previous approach. This has led to significant improvements when performing cell culture media and supplement screens to assess for protein quality. It's now possible to screen directly from small volume, deep well cell culture plates. This technology allows for screening of protein quality attributes at a much earlier stage of cell culture, such as the clonal selection stage. Relieving the cell culture bottleneck will enable us to deliver faster screening of media and supplements to ultimately provide an optimal formulation for our customers' needs. So I'd like to now discuss a case study that we recently performed utilizing the Assay Map Bravo. Here we assess the impact of varying feeds and feed processes on N-glycosylation of monoclonal antibodies produced in CHO cells. So the study was performed in bioreactors, and we were comparing batch versus fed batch processes and the impact they have on N-glycan distribution. Two types of chemically defined, or CD medium, were used in this study: base CD medium A and enhanced CD medium A. For the rest of this presentation, the two types of media will be referred to as enhanced media and non-enhanced media. The fed batch cultures were fed with 5% volume on days 2, 4, 6, and 8. The viable cell densities, or VCD, cell viability, and monoclonal antibody titers were assessed throughout the cell culture time course. Upon monoclonal antibody purification, the N-glycan distributions for each condition were assessed. The viable cell density recorded for the 4 different cell culture conditions are shown up to day 24 of the bioreactor experiment. The blue and red lines show the batch and fed batch data for the non-enhanced media respectively. The green and purple lines show the batch and fed batch data for the enhanced media respectively. Although the peak VCDs show little difference, it's apparent that feeding generally improved VCD in both the non-enhanced and enhanced media conditions from about day 6 onwards. If we look before day 6, there is very little distinction between them. The viability data for the 4 different cell culture conditions are shown up to day 24 of the bioreactor experiment. The blue and red lines show the batch and fed batch data for the non-enhanced media respectively. The green and purple lines show the batch and fed batch data for the enhanced media respectively. Feeding improved viability in the non-enhanced media. This is particularly evident on day 15 when comparing the blue and red lines. There is no clear indication that viability is improved in the enhanced media condition upon feeding when comparing the green and purple lines. The protein titer data for the 4 different cell culture conditions are shown for the bioreactor experiment. The table shows the final titers obtained in micrograms per mil at the point of cell harvest. It is clear for both the enhanced and non-enhanced media conditions that feeding improves the protein titers. So this slide shows the study time points where cell culture supernatants from the bioreactor experiment were collected for each condition. In total, there were 34 samples collected for subsequent protein purification and N-glycan profile characterization. Purification of the 34 samples using the Assay map Bravo was complete in approximately 2 hours. So this slide shows N-glycan profiles of the 4 cell culture conditions at day 14. So there are several observations that can be made from this slide, so I'll just take a moment to describe the figure in a little bit more detail. So again, the x-axis shows the names of the glycan species, and the y-axis shows the percent N-glycan distribution for each of these glycan species. The 2 blue bars show data for the non-enhanced media, and the 2 red bars for the enhanced media. The darker of the 2 colored shadings represents the batch condition, and the lighter of the 2 colored shadings represents the fed batch conditions. It's apparent from this data that feeding decreases galactosylation and generally increases mannose levels. This is evident when you look at the decrease in levels of G1F and G2F upon feeding, and the increase in G0. It's also notable that MAN5 levels are also different. If you look at the G1FA and the G1FB, and also G2F designations, you can clearly see a drop in levels from the dark blue bars to the light blue bars. This is true also of the red bars. There are also differences between the 2 media types. The levels of G0 and MAN5 are higher in the enhanced media compared to the non-enhanced media. Conversely, the levels of G1F and G2F are lower in the enhanced media compared to the non-enhanced media. So this slide shows the N-glycan distribution of the non-enhanced media during the batch time course from day 10 to day 17. In the x-axis are the glycan names, and again the percent glycan distribution are shown in the y-axis. Each colored bar in the chart represents a different glycan species. The major glycan species present in this sample are G0, G0F, G1F, and G2F. Looking across at the data from day 10 through to day 17, there is very little change in the overall glycan distribution. So here we have the N-glycan distribution of the non-enhanced media condition during the fed batch time course. Again, each colored bar in the chart represents a different glycan species. What we've done differently on this particular chart is we've shown the levels of MAN5 as a green line on a secondary y-axis. The purpose of this is to emphasize the change in this particular glycan species over the time course. The levels of MAN5 double over the duration of the time course, from 4% at day 10 to approximately 8% at day 24. Feeding, therefore, clearly has a large impact on the glycan distribution with this particular media. So here we have the N-glycan distribution of the enhanced media condition during the batch time course. Again, the levels of MAN5 are represented by a green line on a secondary y-axis. The levels of MAN5 increase over the duration of the time course, particularly between day 7 and day 13, from approximately 4% to around 7%. From day 14 to day 20, the levels appear to plateau before rising again slightly to 8%. This particular slide shows the N-glycan distribution of the enhanced media condition during the fed batch time course. Compare this data to the batch condition, the levels of MAN5 are higher at day 10, where they are approximately 8%, compared to the 6% in the batch condition from the previous slide. This again shows small differences between the batch and fed batch approaches. The levels of MAN5 show some increase during the time course, particularly after day 14, where they rise to approximately 11% by day 21. So I just wanted to cover some of the main conclusions from this bioreactor study. The Assay map Bravo was successfully utilized to purify 34 samples from a bioreactor experiment in order to study N-glycan profiles. The purification was quick and efficient and was complete within 2 hours. There was generally less galactosylation occurring with the enhanced media compared to the non-enhanced media. This was evident by the comparatively higher levels of G0 and the lower levels of the G1F and G2F species observed. Feeding also led to lower levels of galactosylation, a trend observed for both the enhanced and non-enhanced media. MAN5 levels were generally higher in the enhanced media condition and increased upon feeding for both enhanced and non-enhanced media. Of the glycan species measured, MAN5 levels deviated the most during the bioreactor time course. The levels of MAN5 glycan approximately doubled in the non-enhanced feed condition and also for the batch-enhanced media condition. This case study has revealed a lot of useful information concerning not only the type of medias used, but the way that they are utilized either in a batch or fed batch approach. Using the data gathered here, we have a more thorough understanding of the impact of our media formulations on the glycan profiles observed for this particular cell line. This may aid in the future design of media formulations or cell culture processes where we look to target that particular glycan profile. As a final wrap-up, I just wanted to emphasize the advancements that have been made to our cell culture screening capabilities as a result of the introduction of the Bravo. The really nice thing about this technology is the requirement for relatively small volumes of sample to achieve purification, and so provides protein for further downstream applications, of which we have focused today on N-glycan analysis. This technology allows for screening of protein quality attributes at a much earlier stage of cell culture, such as the clonal selection stage, where high-throughput screens are commonly employed to seek the clone with the most optimal growth and production profiles. The selection of the best cell culture media based on growth and production profiles can now be extended to include desirable protein quality attributes. Taken together, this technology helps implement the optimization and tailoring of cell culture media and supplementation to a customer's specific needs. And finally, I'd like to acknowledge some of the key contributors who have dedicated a lot of time and effort to the work presented here today. Thank you.
Thanks, Ronnie. That was a great presentation. You showed us all some great new methods to improve glycan analysis and how the Assay Map Bravo can begin to help automate high-throughput workflows. Before we start the Q&A session, I want to remind everyone again that this is your final chance to submit questions for our presenter. It looks like we've gotten some great questions already, but I'm encouraging the audience to keep them rolling in. All right. Looks like we have a bunch of good questions, so let's begin the Q&A, and we'll try to get to as many questions as possible. All right, everyone. Looks like we have a bunch of great questions already, so let's get rolling and see if we can get to as many as possible. So, Ronnie, our first question, one of our audience members, actually a couple of them, would like to know if they could get a copy of your monoclonal antibody purification protocol and the N-glycan isolation labeling and identification protocol.
Yeah, absolutely. We can definitely provide those. If you could just respond with a question, and then we can get your contact details through that, and we can send those protocols over to you. Absolutely.
All right, great. Thanks very much, Ronnie. Like I said, like Ronnie said, if you just type in your Q&A box that you would like a copy and send it on over to us, we'll make sure that you get a copy of that. So our next question, one of our audience members would like to know, they say, in the presentation, the ability to screen for protein quality at the clonal stage is mentioned. Can you expand on the benefits of this capability?
Yeah, that's a good question. So each cell clone is genetically predetermined to have a certain metabolism or a protein quality output. So in a sense, one clone can differ quite significantly from another. So for example, a certain clone may be lacking or might have a reduced activity of a key enzyme in the glycosylation pathway. One such enzyme could be galactosyltransferase. So in this instance, that particular clone will produce glycoproteins that lack terminal galactose residues, and so it would be hampered for this particular clone. So it's important if you have the ability to select a clone with the desired protein quality attributes at the clonal selection stage, so you don't have to spend a lot of time and effort optimizing this through cell culture processes or by changing the media composition.
All right, great. Thanks, Ronnie. Next question, one of our audience members asked if you've used the Assay Map Bravo to purify protein for other downstream protein quality assays.
Yeah, that's a good question. We've successfully used IgGs that were purified on the Assay Map to look at charge variant analysis. This is where we consider the acidic and the basic makeup of the protein. This is a commonly used HBLC charge variant-based approach. We've also looked at protein aggregation with these purified IgGs. This is where we're looking at the monomer, dimer, and higher order aggregates of the protein state. Other applications that we're working on include LCMS workflows with peptide mapping and middle down analysis with the IDES enzyme. So yes, there are a lot of downstream protein applications that we're currently using.
Great. Next question. For the N-glycan screening assay that you discussed, What is the minimum volume and concentration of cell culture media required for each of the screening samples?
Yeah, so rather than thinking in terms of volume and protein concentration of media, it's better to really think of it as the amount of protein that's loaded in micrograms. So for the glycan preparation that was described in the presentation, we require a minimum of 50 micrograms. So if you take into account some of the losses that you have during purification, it's best to start with around, I would say, 100 micrograms of load glycoprotein for purification. So this can be either, a small volume of highly concentrated IgG-containing media, such as 100 microliters of 1 mg per mil, or a larger volume of less concentrated IgG-containing media, such as 1 mil of 100 micrograms per mil. Just as long as the load volume doesn't exceed 1 mil, because that's the maximum load volume capacity for the Assay map Bravo.
All right, thank you, Ronnie. Another audience member would like to know what culture conditions most directly, whether positively or negatively, alter protein glycosylation.
Yeah, this is a good question. So there are several variables that can affect glycosylation. Each cell line is different, so it really depends on your cell line and the protein that's being expressed. But there are several instances in the literature that I can draw from, including dissolved oxygen content. As I mentioned, it has a variable effect on glycosylation, and it can be cell line specific. The pH is an important variable. This affects galactosylation and silation. Low temperatures cause a general decrease in sialic acid, and that's also been reported. When we actually talk about cell culture media components, there's also several that can be highlighted from the literature. Low levels of glucose and glutamine lead to generally lower levels of glycosylation in general. Manganese actually has been shown to improve galactose N-terminal galactose species. In terms of silation, there are a couple of modulators of that include DMSO, which decrease silation, and glycerol, that enhances silation. And more recently, there are a few sugars, including sucrose, which have been shown to increase mannose and decrease fucosylation. So there are a lot of media modulators out there, and there's also variables in the cell culture processes, but it really does vary from cell line to cell line.
Great, thanks, Ron. It was very detailed explanation, so we appreciate that. Next question, pretty general question, but we have a few audience members that would like to know, how long is the automated method?
I think that question is probably referring to the glycan preparation part on the assay map. So that can be done within a day. So that whole process from the purification to the isolation on the Bravo, which is all semi-automated, can be done within a day. If you compare that to the manual or the benchtop method that was described in the presentation, that's over 2 days. So there's a considerable time saving with the glycan purification on the Bravo.
So another related question, looks like it'll be our last question, is how long do you need to purify 96 samples with the automated system?
So the 96 samples can be purified all at the same time because there's 96 individual barrels for the assay map head. So the only limitation really is the volume of culture supernatant that's being loaded. And so depending on the load volume and the load flow rate, which can be adjusted on the Bravo, typically takes between 2 to 3 hours. But there is a bit of a window in there depending on those factors that I specified. All
right, great. Thanks, Ronnie. And with that, we've come to the end of our webinar. So So I'd like to remind everyone that the webinar will be archived for 6 months on our website at www.genengnews.com. So if you missed parts of it, you can watch it again, or feel free to forward the link to your friends and colleagues, which we always recommend. I'd like to thank Ronnie again for his informative presentation, and I'd like to thank the audience for their attention and thoughtful questions. And a very special thanks to Agilent for sponsoring this webinar. So hopefully we'll see you again at another Gen webinar in the near future. Goodbye for now.